So the speaker today was going to be Rick Hamasino, who would talk about the plant analog with model organisms to the work that Paul Williams talked about. But it sounds like Rick may not be coming. So let's instead talk about the discovery of niacin. This is one of the things that put Wisconsin on the map 50, 60, 70 years ago, and it's still an important piece of science to know about. So I'm going to try it in about 9 steps to tell you about how niacin was discovered, what that meant as far as what it meant to patients and to the population in general. The first thing that we'll talk about, we already mentioned in class before, and that is the discovery of vitamins in medicine. They were discovered in several places, Madison being one of them. And they were discovered because of a paradigm shift. Instead of looking for the analytical content of a food, there was a biological assay that allowed the detection of very much smaller quantities of very powerful things, which came to be called vitamins. Those vitamins, partly with the work that was done here, with McCullum, Steenbach, Hart, and Babcock, were distinguished one from the other. One group that was soluble in organic solvents, the fat-soluble vitamins, which turned out to be A, D, E, and K, and the water-soluble factors, vitamins, B1, B6, B12, and so on. The development of a paradigm for bioassay for vitamins led people all over the world to take off after these things. There was a race in 4 or 5 major international centers, and one of the races was to discover the cause and the cure for a disease that was very common in the 1910 to 1920, 25 period in the south of this country, Pellagra. The search for the dietary element that would cure Pellagra led to the discovery here of the vitamin niacin, and that in turn immediately led to the cure, essentially, for all those patients who had Pellagra. If there's time, I'll talk a little bit about the cure of another disease, goiter by iodine, but there may not be. Here's a dairy barn, the way it looked in 1910. In the barn here, the single grain experiment was done. Okay, let's see if we can get a big picture. Thank you. The single grain experiment took place in this barn, and it's a national monument of some sort now. It can't be knocked down. It looks as if nature is going to take it down pretty soon, but I hope not. Here's Stephen Moulton Babcock. In front of his balance, sort of symbolizes the old paradigm, which was to look for stuff, proteins, carbohydrates, lipids, in food as a measure of its goodness. But it was Babcock whose intuition led to the experiment we call the single grain experiment, and it changed the paradigm for examining the quality of food from the chemical paradigm, where you just tried to tot up what was in there, and the assumption that if you got enough protein, carbohydrate, and fat and calories, the food was good. They did this experiment with heifers. The experiment lasted for 2 years, and you've heard from me the basic outcome of that experiment, which was that the definition of a good food had to be revised, and the definition of a good assay for nutrition had to be revised too. And now people instead used an experimental protocol in which an animal was made essentially malnourished for some substance, and then that malnourished animal was supplemented with various potential sources of the good nutrient. Those sources were each assayed with the biological assay. The very, very slow purification took place as we found which fractions had the better activities. And eventually these experiments are what led to the discovery of one after the other of the vitamins. Babcock is usually given credit for having been the one who was the brains behind this experiment, but the ones who actually were in there with the animals were Hart, who was the professor, McCullum, who was, I think, probably what we call an instructor today, and Harry Steenbach, who was a graduate student. The three of them, under the tutelage of Babcock, went after these assays and concluded that there was something in there that could only be detected with a biological assay, and changed the whole way of thinking about nutrition. At the same time that this was going on, the experimenters who were working with the heifers realized that this is not going to work. They had to spend money to actually raise large numbers of heifers, enough heifers to get decent statistics. And so McCullum had the idea of using rats. He obtained from a Chicago pet store a set of mating, white, docile, sweet rats, unlike the ones in the barn. And they became the predecessors for most of the white rats used in research for a long time in a lot of places. They allowed one to work with much larger numbers of animals. They had much shorter gestation periods, and so you can see you can go through several generations in much less time than you needed with the with animals as big as a heifer, and it therefore sped up tremendously the ability to test things for nutrient materials. In 1913, this sort of uninteresting title came out, The Necessity of Certain Lipids in the Diet During Growth. This is the paper by McCullum and Davis. Davis is a woman who's credited too little with the work that she did here, but she was a close partner and I think full half of the team that did this. She eventually earned a degree in nutrition and was a professor, I think at Berkeley. Do you know about that, Rod?
I don't know.
Yeah.
I think she ended up with the company.
Maybe so.
Yeah.
But in any case, she never got the kind of credit that he did. So, typical situation for those times. Here is Davis in the middle. And oh, I see what's going on. Okay. I know how to fix that. So. Yes. So here we have McCullum, the young man, Davis, long after the experiments as an older woman, and another volunteer who worked on these nutrition experiments, Cornelia Kennedy, who also made a career for herself in nutrition. So I say here the rest was history. People picked their interesting disease or picked their interesting dietary component, used the bioassay with rats or similar small animals to search for factors that would cure or eliminate one of these nutritional problems. And this didn't take place just here, it took place all over the world and more or less simultaneously. So there's debate about who did what when, but the clear facts are that McCullum discovered vitamin A here. There was a very important step made in the understanding of vitamin D here. The discovery of the water-soluble vitamin niacin took place here. It was confirmed here. Pyridoxal phosphor, pyridoxal, another vitamin, was discovered and developed here. And then the rest of these things are all materials, dietary factors that were in some way or other touched by somebody from the University of Wisconsin. So there was a collection of people who essentially worked on almost everything that you read in the back of a vitamin bottle now during the period from sort of 1913 when the race began until about 1940 when most or all of the vitamins had been detected. They took place at Yale, Rothamsted in England, Utrecht had a nutritional department that was important, Cambridge in Britain, and others as well. If you walk around the campus, you see all of these signs that were placed by the UW Foundation that talk about research results primarily from the Ag School. And those include discovering the vitamins and trace minerals, discovering vitamin A and B, or distinguishing A and B. This is the understanding of the effect of light on ultraviolet, on vitamin D. The trick needed to solve the problem of iron deficiency, the discovery of niacin that cured pellagra, and the discovery of a simple way to enrich table salt so as to make goiter, essentially, that didn't occur any longer. Well, so we tend, out of our local pride, to talk about Wisconsin as if it's where the whole thing took place. But the truth is that vitamins, or something by another name that with the same qualities, were suspected or established long before here. For example, you all heard about the story about scurvy. Seamen who went on long travels got the disease called scurvy, in which their gums receded, their teeth were loosened and lost. They became severely ill and actually died from this disease, which could be completely cured by having simply a little taste of a lemon or a lime or any of the citrus fruits. And this was as early as 1753 that one of the people who watched this in the British Navy, Lind, published the fact that you could solve scurvy by just carrying kegs full of fruit juice. The British sailors came to be called limeys, and it was because of the lime that we took along. Osborne and Mendel, a pair of nutritionists at Yale, worked in almost complete parallel with the people here. And I looked at the letters exchanged between Babcock at the time and the people at Yale, and there were some hard feelings about who did what first and who deserved credit. But clearly, it was not a solo effort. There were several groups involved in these things. Similarly, beriberi, the disease discovered and studied, I should say, by Eichmann. This, I don't have a date here, but this is well before the date of 1913 when the vitamins were discovered, quote here. Casimir Funk in Europe characterized components of the diet. He called them vitamines, vit because they encouraged life, and amines because he thought that all the vitamins were going to turn out to be amino-containing groups. He was wrong about that. Hopkins in Britain had published papers well before 1913 saying that there were diseases that were caused by nutritional deficits. And so we should be careful when we claim that Wisconsin was the cradle of the vitamins. It certainly is true for vitamin A and for niacin. It's less so and not at all so for some of the other ones. Pellagra in the American South was so common that it became a major focus of the public health service during the period between 1900 and 1920, I'd say. The occurrence in orphanages and prisons correlated, of course, with the lousy diet that was served at prisons and orphanages. People who looked a little more carefully at the diet had the impression that it was the corn in the diet, cheap, easy corn, that was causing the trouble. There were several hypotheses about how this might take place. One was that maybe corn had something in it that was poisonous, and if you had too much corn, it caused the disease pellagra. The other possibility is that there was something that's essential in the diet, but not present in diets rich in corn. And then you'd have to cure the disease by finding the essential factor. And there was also strong belief in some quarters, including the American Medical Association, that pellagra was actually a communicable disease. You got it from somebody else. Goldberger was the man sent out by the Public Health Service to try to solve this problem. And it's really a great story that I'd love to talk at length about, but in short, Goldberger essentially invented the epidemiological method of examining diet. He went to places where pellagra was common, not just the prisons and the orphanages, but to small towns, cotton mill towns, where it was also fairly common. And he went to family after family after family, asking each member of the family exactly what they ate and how often and when and with whom. He went through the description of the work that they did and who they worked next to and what the materials were that they worked with. He did an incredible piece of epidemiology, practically single-handedly. And he reached the conclusion, which was right but not readily accepted, that the problem was something was missing in the diet. Slowly, over time, the other side gave up, but it was a real struggle. This guy was a real hero. He fought hard to make his hypothesis understood and believed. So here was the story. The disease has the symptoms shown on the left: diarrhea, dermatitis, dementia, and finally death. There was a time when in both prisons and orphanages, half of the residents had pellagra, and some 30 or 40% of that half were going to die from pellagra. It was a very serious disease. It was sometimes called the butterfly disease because one of the first things that happened was the cheeks turned red and rough, and it sort of had the shape of a butterfly there. And it turned out that by dumb luck, a veterinarian figured out that there was a disease in dogs that resembled in every way the disease of pellagra in humans. So the disease is called black tongue. I don't actually know how the vet figured this out, but it certainly was incredibly important to what went on here. Is this thing dying on me, or? Yeah. Oh, I see. All right. Well, I'm glad. So Joseph Goldberger from the U.S. Public Health Service was one who did these studies. He was so convinced that this was not communicable that he and his wife went through an experiment that no regulatory group would ever allow now. They actually held what they called a filth party, where they ate and injected into themselves snot, scabs, blood from patients who had pellagra. This is the definition of a devoted wife, I'd say, to put up with this. But it, of course, turned out that they did not develop pellagra. And they won the argument with the American Medical Association's Thompson-McFadden Commission. There he is. Well, so the fact that black tongue was an analog, or thought to be an analog of pellagra, allowed people to use dogs as their experimental animal. And this question of what caused pellagra was taken up in Madison in the early, I think probably the early 30s the work started. The animals were kept in the attic of a building that still stands across from here, here. When I first came to Madison, that attic where the dogs were still had bones from various animals that had been subjected to dietary manipulations. These two people, Conrad Elvium and Frank Strong, were the driving force behind the discovery of niacin. They had graduate students, Madden and Woolley, who co-published this paper, and both went on to very distinguished careers in nutrition, but the credit for this is usually given to Elvium and Strong. Strong was an organic chemist. Elvium was a nutritionist. He eventually went on to be the dean of the graduate school and the president of the university, but he knew his experimental protocol for nutrition, and between the two of them, they eventually showed, with the help, lots of help of their graduate students, that niacin, nicotinic acid, very simple, cheap compound to make, completely cured black tongue, and within a few days, in those days you didn't need to do clinical trials, within a few days niacin was being given to pellagra patients and cured them completely. So the statement in this simple paper came out in 1937 reminds me of the statement that came out in the 1950s paper by Watson and Crick. These two, or these four, say, the observation that a deficiency of this material may be the cause of black tongue is most interesting. It's like saying it has not escaped our attention that this mechanism suggests a replication mechanism. Very modest thing. One of the controversies that I've read about in the literature and in the personal notes of several of the people who were involved in this controversy is who actually deserves the credit for this. And there's a series of interesting back and forth things, unpublished letters that I've looked at, in which the question is, if you are an organic chemist and you have the equipment to test the compositional analysis of something, and somebody comes to you with it, do you get credit for the discovery if you just do the compositional analysis? And so it boiled down to this. If somebody says to you, is this stuff niacin, and you as a chemist say, yes it is, then you just confirmed what he did and it's his discovery. If you say to the chemist, what is this stuff, this pure stuff, and he tells you, then he's a co-discoverer. And there was a lot of ruffled feathers over this issue. This instrument is the instrument that K.P. Link had in his laboratory. He had the only one on the campus, and it was a combustion train that was capable of determining the compositional analysis of large quantities, milligram to gram quantities of material. One of the things that it's hard for us to appreciate now is how difficult it was to do work with very low concentrations or low quantities of stuff. Vitamins, because they become cofactors and get circulated, reused, we don't need very much of them. And so the concentration of vitamins in tissue is orders of magnitude lower than the concentration of fuels, for example. And some of these things had to be discovered by monumental experiments with very large amounts of starting material. And it's no wonder they weren't discovered earlier. While I was reading the papers about this period, I discovered that the Nobel Committee had changed their policy. The policy used to be the background papers that come to the Nobel Institute are pure. Nobody ever gets to see them. But they've actually changed the policy so that now, after 50 years, the stuff that's been accumulated in the files of a Nobel laureate is open to the public. And I have here two things. One, a letter nominating it's hard for me to read, but this is a nomination letter nominating Conrad Elvium for the Nobel Prize. And when I looked carefully, I discovered that there had been a whole lot of nominations of Wisconsin people for Nobel Prizes. Each of these was one group that nominated. Sometimes in the same year, two groups nominated. But look at this. Elvium was nominated 10 times or more. Frank Strong was nominated several times. The students, Madden and Woolley, were nominated. McCollum was nominated. None of them ever actually won the Nobel Prize, but they certainly were at the tippy top of their discipline, and it's interesting to wonder why they didn't ever quite make it over the threshold. When I went through K.P. Link's personal papers a couple of years ago, I discovered that he'd kept a file called Pre-Nobel. And in it went everything that was related to the Nobel Prize. If he had a letter from somebody with the prize, he put it in there. It was clear that he thought about this. Here are the prizes that were actually awarded, Nobel Prizes, and what you see from this is that for a period between about 1910 and 1940, a lot of the excitement in the life sciences, in medicine and in chemistry, was in the discovery and the identification and the mechanism of vitamin function. So what I set out to do was 9 things, and I think we've done that now. The essential factors were discovered here. The paradigm shift was pushed by the discoveries here. The A and B groups were discovered and named here. And then the work took place all over the globe after that. It's hard to attribute success to any one group or person. I want to turn now to something else that was going on at the same time. We have time. And that is the biochemical studies of metabolism. In the 1910 to 1940 period, a lot of the pathways that we use to degrade glucose, for example, to get energy from it, were being explored by biochemists. They had developed a few tools, one of which was a manometric technique for measuring oxidases. It was called the Warburg manometer. And with those, I think, quite crude tools, it was possible, they were able to take apart the pathways that degrade glucose to pyruvate and other related pathways. And in the course of doing that, people like, well, first I should say Buchner, who won the Nobel Prize in 1907, was the one who discovered you could take yeast. At the time that he did his experiments, the belief was that you couldn't get fermentation, you couldn't convert glucose to ethanol except in a living organism. And this sort of meant that living organisms had some kind of magical stuff in them that the dead stuff didn't have. Well, Buchner developed a technique for grinding up yeast cells so completely that there were no living cells left in this grindate that he made. And he discovered that if he added the right stuff to this ground up stuff, that it would actually ferment glucose all the way to ethanol. And that meant that one could start looking into the broken cell extracts to take apart the individual components that were responsible for the series of steps that degraded these things and start characterizing each of them as a chemical entity. Otto Warburg was the one who deserves credit for most of this, but there were, of course, many people at the same time doing these experiments. But the critical thing from the point of view of the lecture today is that in the course of purifying these enzymes that break down sugars, it became clear that there were enzymes, which were proteins, and coenzymes, which were small molecules, and the enzymes wouldn't work without the coenzymes. It turned out also that the coenzymes, like enzymes, didn't get used up in the process. They were recycled. And that, of course, accounts for the fact that they are needed in such small quantities. So here's Otto Warburg. There is niacin on the left. I don't know if I can point to the... Yeah, here, this is the business end of the cofactor NADH, or NAD. And it's that compound that's derived from this one that makes the vitamin essential in the cofactor. This is nicotine, which doesn't have any serious it's not biologically similar and it's not chemically very similar, but the name sometimes suggests that it is. Okay, so by 1940, the search for vitamins was pretty much concluded. There have been surprises since then, but the main categories of coenzymes and vitamins had been discovered by 1940. There was one more set of experiments that's worth noting along with these, and that is the search for things that were inorganic and were necessary in the diet. The way that that search was conducted was very much like the search for the organic compounds, but in the beginning, one took a material that might be capable of supplying the needed mineral and ashed it with strong acid so that there was nothing left organic, just the ashes, and that would be the salts. And the search for activity in the salts led to the discovery of several requirements for inorganic components, one of which turned out to be iodine. In the Midwest, before the turn of the 20th century, goiter was actually a fairly common disease. And this guy is an extreme case of goiter, but what's happened is that his thyroid gland, which is here, is trying to make thyroxine, but it can't do that without iodine because thyroxine contains iodine. So people who live in areas where there's not a ready source of iodine in the soil or in the sea sometimes develop this disease. The thyroid gland tries hard to make thyroxine, gets bigger and bigger and bigger, and the individual nonetheless suffers from the lack of thyroxine, which is metabolically important. This is a picture of thyroxine, and circles all the iodine atoms that are in it. It's clear without iodine you can't make this stuff. Hart and Steenbach actually collaborated in a study that showed that the cure for goiter is iodine. And again, iodine is a dirt cheap component, so that the effectuation of a cure was extremely inexpensive, just because the material needed was cheap. There were, at the time that I wrote this, 2 billion people in the world that are actually deficient in iodine for reasons that I mentioned. Either they live away from the salt sea, or they're in places where the soil is very poor in iodine. And this statement shocked me when I saw it, but I actually read a little bit about it, and it's true that the leading preventable cause of intellectual disabilities is the lack of iodine. A thing is simply cured as simply putting a few cents worth of iodine into something that you eat. When you buy salt now, you always buy iodized salt. And the contribution from here was that E.B. Hart, the professor that led the single grain experiment, figured out a simple way to stabilize iodine dissolved in or associated with table salt, and essentially wiped out the problem of goiter. Now, just a few words about what happened to the people who I'm talking about here. Here's a painting by Aaron Bohr Rod of Conrad Elvium, who progressed from professor of biochemistry to chair of biochemistry to dean of graduate school to president of the university, and he died. He literally died in office. He keeled over in his president's office. He was prodigious in the training that he gave, in the papers that he published, in the number of people who came from his laboratory and then ran major laboratories of their own. And to accomplish this while you're spending half your time as the dean of the graduate school is a substantial accomplishment. This I have included just to remind you that of all those things that are on the back of a bottle, most of them have been touched by somebody here. And this is a picture that if you haven't seen the real thing, I recommend that you do so. This is a mural by John Steuart Curry. It was painted just in the first years of the Second World War, at a time when the Wisconsin work on vitamins had pretty much slowed down. They'd been discovered. Curry was an interesting guy himself. In the College of Agriculture, he was the visiting professor of art. And you figure how that happened. But it turned out to be a wonderful combination. Curry went around the state for a whole year, talking with rural groups, talking with individual farmers, artists, encouraging them to make their own art. They developed groups all over the state of local art colonies. They published a wonderful book of rural art, all because of his serving here as the ag school's visiting professor of art. But another of the things that he did was he painted several murals in Madison, and if you haven't seen them, you really should try to do so. This is a mural. The size is actually bigger than on the screen here. It's 9 feet by 13 feet, and it's located in the old biochemistry building, which is straight across the street here. If you wish to see it, it's a little tricky to get there now because of a reorganization of the building, but go in the front door, walk all the way to the end of the hall, and around the corner, and you'll see this. The people on this are interesting. There are several things worth noting. Have I shown you this picture before in class?
Yes, you did.
Okay, well, so here's this little boy with rickets. There's an animal over here with ribs show. On the other side, everybody's happy and healthy. And the scientists that are responsible, that looks like Babcock there, for example, this looks like Hart, are shown, as well as the women who are delivering the care. I wonder sometimes if they might be the 2 women who worked on the vitamin project, the ones who weren't credited properly. Somebody else has pointed out to me that this thing even has soil erosion over here on that hill, whereas there's soil conservation methods here. So Curry actually worked with the scientists here to make this mural. I have pictures in my office of the mural half completed, with Curry on his knees trying to paint and Steenbach pointing, do this. But it's a nice crystallization of a lot of the scientific research that went on here during the early part of the 20th century. There's a second mural that's in the law school's library, a very large mural called The Freeing of the Slaves. And there are around campus in several places other murals. There is a painting, not a mural, a painting, an oil painting of a farmer in the field. Became iconic. It was used in the recruitment of war participants. It appeared everywhere for a while. And it hung behind the dean's desk in the College of Ag and Life Sciences for years, until somebody figured out that that Curry painting was worth $1 million. And the dean apparently decided that he didn't want that anywhere near his office, and it was removed. I've said two things that should have struck you as contradictory. One is that I've said that corn in the diet is bad for you, if you're a human in an orphanage. And I've said that the lack of the corn in the single grain experiment was the best nutrient for heifers. And it turns out that there's an explanation for this. It's not a mystery. The niacin that's in corn is not easily extracted from the corn. Indians discovered that they could actually treat corn, pre-treat corn in such a way that the niacin came out. But if you didn't do that, then it wasn't really available, or not largely available. And as for why cattle need and can use corn, it seems to me that all you have to say is that a cow is a ruminant. There is an extra stomach there that has a powerful collection of digestive enzymes, and maybe that's the answer. Is that sensible, right? Somebody caught me in a lecture saying, well, why is it good for you and bad for them? So this is the nearest. All right, so that was Rick Amasino talking to you about plants. I'm sorry for the mix-up. I'm sure it's my fault, not his. Are there any questions about what I've talked about today? Yes.
I have one comment about iodized salt.
Mm-hmm.
And that is, in today's world of ceremonial sea salt and aerial salt application and so on, there's a push to not use iodized salt. And so goiter is re-emerging in inland countries now in some places, or in inland places. Yeah. And it's with the especially well-to-do who can afford the
fancy
gourmet salt instead of the regular salt.
Well, I thought that seafood from the oceans also is a source of iodine, and so I presume wealthy people can afford imported seafood, and that might help a little. Yeah.
But why then did goiter show up in the Pacific Northwest, where there's...
Apparently the Pacific Northwest has soil which is very poor in iodine. And so the
it showed up with people who lived inland from the Pacific Northwest, still in that area called the Pacific Northwest. So if they were on the coast, they'd be maybe eating fish that had... That's
right. Presumably if you lived right on the coast, at a time when fish could move 50 miles. It's an amazing era, this era of looking for nutrients, and when you read the papers, you realize that it was incredibly exciting to the people involved in it. And when you read the back story, you realize that the human enterprise here, like every place else, involved some crankiness and some unhappiness about somebody else beating you or seeming to beat you. But it was a wonderful era in biochemistry.
You know, you may have, I can't wait, but you may have explained this, but why did these various discoveries of vitamins slow down in the 1940s, at least on this campus? Is it because they had all been discovered, almost all?
Yes. They were essentially, I mean, I don't think I know of any vitamin that was discovered after 1940.
Okay.
Yeah. There are some sort of pseudovitamins. Some simple creatures like bacteria have complicated cofactors, which are made out of precursors that are essentially vitamins for the bacteria. Okay, I apologize again for the change of subject. See you next time.